Hello, everybody. It's nice up here. You should try it afterwards. I'm very glad that I go after you. I'm just going to show you some black and white boring pictures of electron microscopy. So I'm going to introduce the SPOT-RASTR to you, but first, so one of the most important questions we have in science is how does life look like in details, right? So we can just take a look by eye, and I'm using this leaf as an example. Or if we want to zoom in, we can get a light microscope and look into the cells and think like huge, giant things like that, giant in my scale. Or you can push it further to electron microscope and see like some finer details like organelles, giant micromolecules, things like that, to better study all of these shapes and structures that will be useful to study the function and other stuff. As an example, you can think about the COVID virus. So when we realized that this is a virus that's causing that infection and disease, we took a picture of that using electron microscope and we push it to 2 angstrom resolution, and the resolution is our currency in electron microscopy. So based on that, you can then make the vaccines and make drugs, everything. So, which is pretty useful to study the structure. I guess I just wanted to state that. And then there are many challenges during the sample preparation for electron microscope. So you need a lot of pictures first, and then you need all of your protein or sample of interest to be inside a thin layer of ice, and it forms something that we call the air-water interface, which is huge comparing to the size of the molecules, and most of the time they touch that air-water interface, as people call it, the deadly touch, and they get denatured like a rotten apple. Or the other problem the air-water interface creates is put all of the samples or proteins at the same orientation. So if you look at them like from the top, and you see the top view, you may not be able to differentiate between an apple or a red cucumber. I don't know if that exists or not. So these are the 2 main challenges that we wanted to solve. So we chose this SPOT-RASTR, and you don't want to worry about like all these words. So sample preparation and tubes, I'm just gonna focus on that part. The idea is to use lipid nanotubes, and you see the diameter is 30 nanometer, and decorate them with the sample of interest using these lipid tags. So when you have protein with the like defined tag, and you have the catching tag in the other side of the lipid, you can catch your particles. You see a naked tube here comparing to the decorated tube. And then this would be a schematic of this method. So we tried it in different ways, like different methods of electron microscopy, and we realized that yes, actually, in fact, it is protecting the particles from the air-water interface, like comparing to the free particles, because it is embedding them inside the ice attached to the tube itself. If you go back to the same analogy, like instead of having rotten apples or apples from top or red cucumbers, you will see something like this. So different views of the same apple, and with different views, you get high resolution structures and get an exact idea how your molecule will look like. And this is a real image of the sample. So this is the tube, electron microscopy, black and white, sorry about that. But you see the proteins are completely like decorated on every angle. And from that point, we take these things called 2D classification or 2D classes. You see they are in different orientation, kind of. And we get a 3D structure, and we got the 3.5 angstrom resolution, which is fairly good for this, considering we are introducing new method. And then when we compare the particles, angular distribution, as we call it, so different angles of the particles, how many particles in what angle do you have? With the particles that we prepared like with normal sample preparation, and in the middle you see the spot prepared particles, you will see it is completely spread. So we have different angles of the particles because we have this 360 angle of the tube by itself. With that, I'm finishing my talk, and I hope you enjoyed it.